Hello everyone, welcome to my channel Provocative Biofield. I am Sagarika Babu. Today we are going to see about ribosomes, an important cell organelle that is present inside a cell. Before entering into the video, please subscribe to my channel and hit the bell icon to get the notifications of all my videos. Ribosome is a structure found inside the cell that is involved in making the proteins. They are the granular structures first observed under the electron microscope as a dense particles by George Pallet. Hence, these ribosomes are also known as Pallet particles. George Pallet was the first person to observe these ribosomes in animal cell under the electron microscope in the year 1955. These ribosomes are composed of RNA and proteins. As ribosomes are composed of RNA, that is ribonucleic acid, and protein, these ribosomes are also known as ribonucleoprotein particles. Ribosomes are not surrounded by any membrane. They help to link amino acids together to form proteins. Ribosomes reads the messenger RNA sequence and translate that genetic code into a specified string of amino acids which grow into long chains that fold to form the proteins. Ribosomes are found in both prokaryotes and eukaryotes. In prokaryotes, the ribosomes are found in free state, while in eukaryotes, these are either found in free state or associated with endoplasmic reticulum by their larger subunit. In eukaryotic cell, ribosomes are also found in chloroplast and mitochondria. Hence, ribosomes are also called organelle within an organelle. Ribosomes are composed of two subunits. Larger subunit has a dome-like shape, whereas smaller subunit occurs above the larger subunit and form a cap-like structure. These are negatively charged molecules containing rRNA and protein. Both the subunits remain united with each other due to the specific concentration of magnesium ions. Ribosomal subunits are separated when concentration of the magnesium ions reduces below a critical level. During protein synthesis, many ribosomes form a chain on a common mRNA and form the polysome or polyribosome or ergosome. In prokaryotes, the ribosome present is 70S ribosome, while in eukaryotes it is 80S ribosomes. What is S? S is Swedberg's unit, which stands for sedimentation coefficient, indirectly is a measure of density and size. In prokaryotes, 70S ribosome are made up of 30S subunit which is smaller and 50S subunit which is larger. And in eukaryotes, 80S ribosome is present as 40S smaller subunit and 60S larger subunit. In prokaryotes, rRNA and proteins are synthesized in cytoplasm, while in eukaryotes, ribosomal subunits are synthesized in nucleolus, which is a spherical body found inside the nucleus. Two main functions are carried out by the ribosome. One is decoding the message and the another one is formation of peptide bonds. So finally, ribosome assembles the amino acids to form proteins that are essential to carry out the cellular functions. That is why ribosomes are also known as protein factory of the cell. So the major function of the ribosome is it is the site of protein synthesis in the cell. Thanks for watching the video.